Well, it's for me really a great pleasure and a big honor to introduce today our first plenary lecture. Dr. Claire Brown is Associate Professor at the Department of Physiology at McGill University, and she is the Director of the Advanced Bioimaging Facility. Claire is a person that has strong commitment with the developing of community. And she is the co-founder of the Canadian Network in Scientific Platform and Canadian Bioimaging, and is the representative of Canadian Bioimaging in global bioimaging as well. Claire is also co-chair of the North American Bioimaging Initiative, and in the last 25 years, she has devoted a lot of her energy in the different areas of research in bioimaging. And I think today, with this talk from Biophysics to Imaging Platform to International Network, she will cover all this tour. So thank you very much. It's an honor and a pleasure, as I say there.
So thank you to the organizers for the kind invitation and Lionel for the kind introduction. What I'd like to do today is give you a little bit of a tour. So I decided for the lecture today, I'm going to give you a little bit of a story about my career path, but sort of filter in aspects of technology development that I've experienced through my own research. And so I'd start with I've always loved science from when I was a little kid. I was always interested in that, signing up for the extra programs after school to do experiments. And in undergraduate, I did physical chemistry, and I'll tell you a little bit about that project. In graduate studies, I moved into more cell biology and correlation microscopy, where I'll explain a bit of a link I have with Lionel. And I'll talk a bit about a postdoc I did at the University of Virginia, applying these techniques in cell biology, which really led me to my current position at the Advanced Biomaging facility at McGill. And then later in my career, moved more into these collaborative networks. So I started my undergraduate in chemistry, and I always preferred chemistry to biology because you could figure things out. So if you got to the exam and you forgot something, you could work it out. You didn't have to memorize the whole life cycle of every worm that is on the planet. So I ended up in physical chemistry, which was more towards the maths and things that I liked. And this was a really interesting project for my undergraduate honors, where the researcher I was working with my cells, which form tubular structures at very precise concentrations. And they were putting this surfactant into the cooling system at the university in the library. And when these tubular micelles formed, they completely eliminated the turbulence in the water and actually reduced the cost of pumping the water through the cooling system by 95%. So they could use a gallon of this soap basically and put it in. And I thought it was quite interesting. Now when I look back, it's very similar to a cell membrane. And my job was to look at the electrochemistry of these molecules and see how they impacted the piping and potential cause corrosion and whatnot. And of course, knowing much more about biophysics now than I did then, we found out that they actually helped eliminate corrosion by putting a coating on the inside of the piping, and were actually protective. But when I was coming to the end of the project, I was actually finding it a little bit boring. I was thinking, if your pipes rust, then you can buy some new pipes. And I was wanting to move a little bit away from pure chemistry. So that's where biophysics came in. So I found a wonderful research lab at Western University in London, Ontario in Canada. And my supervisor was interested in applying quantitative biophysical tools to cell biology. And I wanted to bring in my microscopy experience here. This is a Biorad MRC 600, which was actually the advanced model. We only had the 500. And this is what I did all of my PhD work on. And just for the early career people in the audience, I wanted to point out a few things. This was before there was GFP, so everything was done with antibodies and everything was on fixed cells. And we had this was the confocal for the university. So we had to compete with anatomy and cell biology and biochemistry. And so I would often do my work on Friday evenings or Saturdays or holidays. And I would love it because you could go for the whole day and nobody would bother you and get all of your data. And the most interesting thing was, if you see on the image here, these are the filter cubes. They're filter packets, really. They're not even cubes. And you actually, when we did 2-color imaging, we only had one channel. So we actually had to put the green cube in and take a picture and take it out, and put the red cube in and take a picture. Then you take your red picture for the next frame, take it out, put the green one back in, and do this back and forth for all of your imaging. And the other thing I always like to tell people is we had 8 digits to name our files. So you had to be creative there as well. So this is what the cubes actually look like, filter blocks, that's what they call them. So you can see that, very similar to what you would have with a dichroic. And the confocal dye was really designed to get a pretty picture, which I think we still see nowadays in a lot of the newer marketing of instruments is how to get a really beautiful picture. But we were really interested in quantification. So I had read in the manual that the confocal is only as good as its alignment, but our technician would not let us touch anything. So I snuck in on a Saturday and got the manual and started reading about how to align the pinhole, and learned how to do that and showed everyone else in our lab. I think the technician was okay about it after. So we really had to learn, we almost had to deprogram the manual and the microscope so we could be quantitative. And I don't really have time to go into a lot of detail of all the different techniques, but I just wanted to mention correlation spectroscopy because I think it's a fascinating tool, and it also gives me a common link with Lionel. So the way fluorescence correlation spectroscopy works is you have a focused laser beam, and you basically just sit it there and the molecules move in and out of the beam. And if you have a very dispersed distribution of your molecules, then as the molecules move in and out of the beam, you have these intensity fluctuation. But if you have a cluster of molecules where, say, you have 3 or 4 fluorophores instead of 1, then these fluctuations have a much higher amplitude. So if you do a statistical analysis of looking at this signal for over many seconds or minutes, you can actually get information about the aggregation state of the molecules. In addition, you can look at the shape of these fluctuations. So if the molecule is moving through the beam fairly quickly, you get a very narrow fluctuation. And if it's a larger molecule moving through more slowly, you get a broader fluctuation. So between the amplitude and the shape of what's called the autocorrelation function, which is basically just a statistical analysis of thousands of these events, you can measure things about the distribution and the dynamics of molecules. And this was originally developed to do in solution. So people would put a dye molecule in solution and measure the diffusion constant and the concentration. But in our lab, we were interested in applying this to cell biology. So we moved into a method of looking at, instead of waiting for the molecules to move, we would use the confocal microscope and move the laser. So the sample was fixed, and by moving the laser across, we could still calculate this autocorrelation function. We didn't have dynamic information because the sample was fixed, but we could get information about the distribution of the molecules. And this was a project we did in collaboration with Tel Aviv University. And you can see here one of these correlation functions, and we would fit the amplitude, and it would tell us something about the protein distribution. And this is actually a figure that was in my PhD thesis, where we had a wild type protein, which was just sitting in the membrane. And then we had a protein with it was actually a single amino acid mutation here to a tyrosine that led to an internalization signal. And you can visually see the protein is much more clustered, but we were able to actually quantify this biologically and also see that it was temperature sensitive. So if things were at 4 degrees, they didn't aggregate. And one thing I'll also point out is on this very basic confocal, we were getting quite beautiful images, I think, even at this time. So then we went on to do image cross-correlation microscopy. And this is another very powerful tool where you now look at 2 molecules. And if the molecules are moving through the laser beam at the same time, then the fluctuations are coincident. And if they're moving through separately and randomly, then they're not. And you calculate now what's called an image cross-correlation function. And the amplitude is related to the concentration of molecules that are interacting. So we applied this to the same system, but now we were trying to show that these molecules were going into the cell through clathrin-mediated endocytosis. And of course, again, remember, there's no live imaging. Nobody's watching clathrin and endosomes pinching off of cells or any of that. So So we were able to do 2-color imaging now, where I was changing those cubes manually, and show that when we had this internalization sequence on the protein, it was now colocalized with clathrin. And again, we're able to quantify that. So when I finished my graduate studies, I was very happy to accept a postdoctoral fellowship at the Institut Curie in Paris, France, where I was working on multiphoton microscopy applied to cell biology. And I was kind of going to jump right from my postdoc to my PhD to my second postdoc, because this was a bit of a messy period in my life. And then I was thinking about the young students here today and the early career researchers, and thinking, you know, these are the important things we need to talk about. And I think too often we show the superstar story in our presentations and show the perfect data. And so I just wanted to share a little bit about this. It was a difficult time. I actually had an unexpected pregnancy and a welcome surprise of my son. So there's a couple of pictures there, one when he's young, and then some of us when he was young. And then that other picture was right after the LABIMeeting, actually, in Dublin in Ireland. He came over to meet me there. And I always point out to people that I am standing in the picture. I'm not sitting, but so he's quite tall. So it was a surprise, and I ended up bringing him up on my own. So I've been a single mom to him. He's 22 now. So that created a lot of uncertainty in my career path and so on. I decided to I took 5 months maternity leave, and then I took a position teaching chemistry for a year at a small university in Eastern Canada. And that was an interesting experience. It was a good job, but it also told me I didn't want to be a teacher. So I like to teach a bit, but I really knew I wanted to go back to the research. So I found a job at SUNY Upstate University in Syracuse, New York. And part of the appeal there was that I was close to the border where my family was. So I could come back on weekends and spend time with my sister and brother who lived near there. And I moved about 10 days, I guess 2 weeks before September 11th. So when that happened, the border shut down and it was extremely hard to go home. And I felt a little bit isolated. And then I started to realize my supervisor was a little bit crazy. So I was supposed to go to his lab and build a fluorescence correlation microscope. And I was very excited because I wanted to bring a new tool to my skill set and learn how to build instruments. And when I got there, I quickly found out that he had no idea how to build instruments, and I wasn't going to learn how to build instruments from him. And then I also found out he was I thought he was manic depressive at the time. But shortly after I left the lab, I found out that he was arrested for drug possession. So he was actually having some issues there. So he ended up being dismissed from the university shortly after I left. But this was a really difficult time. My son was only 1.5 years old. I had just moved to the US. I had found a place to live and a daycare, and now I'm picking up and moving again. But the positive side of the story is I ended up going to Rick Horwitz's lab at the University of Virginia, and he had just started a collaboration with one of my co-lab mates, Paul Wiseman at McGill University, on correlation microscopy. And I read the job ad, and it was me. Like it was one of those things that's like, oh my gosh, they want me, you know? So I contacted him and told him my situation, said, you can't call my supervisor. You have to trust me. I'm a good student, you know? And I ended up moving down there for about 4 years. It was a great place to live. And they had this huge $10 million NIH grant for the Cell Migration Consortium. And that was really where I started to really learn live cell imaging and how to apply what I was doing to living systems. And for those of you who know or will maybe meet Nikki Biley in the next few days, she's the program coordinator for Biomaging North America. That's where I actually had met her. She was the program administrator for the Cell Migration Consortium. So I was involved in recruiting her to Bioimaging North America. At the same time, I was part of the imaging group on this NIH grant, and Enrico Graton was the lead of that part of the grant. And he's at University of California, Irvine, and was Lionel's postdoc supervisor. So we have a connection there. And then Michelle Dignan came into the lab about 2 years after me. And so she came to Virginia and learned what we were doing with the cell biology there, and we were connected. So it's interesting to see how life goes. And through a really difficult situation, I actually ended up in a really great lab, and really sort of set off my career into platforms. So just a couple snapshots from that time. The lab was focused on cell migration and adhesion. So this is a movie of Paxillin, which is a cell migration, an adhesion protein that we've been quite interested in. And it really regulates the adhesion of cells to the surface and the movement of the cells. And this is really important for understanding normal physiology like wound healing and the immune response. And it's also very important for pathologies like cancer and neurological disease. So in the Horwitz lab, I was tasked with applying these correlation techniques now on living systems. So expanding what I had done in my graduate work on fixed cells. And I actually like to put up this slide because I use the quote, "Seeing is not believing," because the joy of correlation microscopy is you can see things that you can measure things that you can't see. So we did this great experiment. This is still one of my favorite experiments for quantitative imaging. This is a membrane anchored GFP without any kind of function to it. And then this is cells with an alpha-5 integrin, which is one of the main molecules that binds the cells to the cover slip. But they're on polylysine, so the integrin is not activated. And then this is the same cells with the integrin, but now on fibronectin, which activates the integrin and causes clustering. And I don't know how good the image is up here. You may be able to tell there's a difference, but I don't think you'd be able to say much about the protein. So we went in and used image correlation, and we were able to actually show that in the case of the fibronectin, the clusters had about 3 proteins per cluster. And our eye is just not sensitive enough to see that kind of a small difference in intensity. And the difference in intensity ended up being about 30%. So if you're in an adhesion, you had about 30% more of the molecule than if you're outside. And you can see a time lapse here of the Paxillin, which is very easy to quantify in the adhesions, and the alpha-5 integrin in the same cell where you don't see any localization at all. And the interesting thing was the reason we figured this out is I was doing frapping experiments, so photobleaching recovery, and I was trying to show that the alpha-5 integrin was moving more slowly in the adhesions. So I would bleach an area where Paxillin was, and then I would bleach an area nearby. And the area where Paxillin was always higher intensity. And so I just noticed that in every one of my experiments. So this is why we went back and quantified it. The other thing we did is based on this monomer, this membrane anchored protein, we were able to calibrate our confocal. So we could actually say how many molecules of GFP we had in each location. And since we have 1 GFP for every integrin, we actually did this experiment where we actually had the number of integrins in each of these adhesions. So you can see the adhesions outlined here, and then we quantified the actual numbers. I guess it would be fun to go back and redo this with some of the new single molecule imaging techniques. But this was in the early 2000s, so it was quite exciting at the time. And here you can see like where these adhesions are in the lower part. They're quite hard to see even with the Paxillin. Or no, this is the integrin. They're quite hard to see, but we were able to quantify it by calibrating the microscope. So the other big advance that happened at this time was raster image correlation spectroscopy. And this was discovered by Enrico Graton. But I always like to give myself a little bit of credit towards that because when I first met him, he was doing only point-based FCS. And they were doing a little bit of work where they would scan the laser beam in a circle. So they could get like 64 or so data points. And I think by working with us and working with Paul Wiseman and through the Cell Migration Consortium, it got him thinking in the spatial domain. And then being I always say he's on a parallel universe to the rest of us. He just went with it, and he's developed so many amazing tools for microscopy that also bring in spatial information. So this is a really interesting concept. The idea was that when the confocal laser beam is scanning across your sample, the molecules are actually moving. And you don't actually see them moving, but if you do the correlation analysis, you can pick this up. So this is actually simulated data. On the right side, you have something that's moving very slowly. So this would look very similar to our integrin images or Paxillin images. And when we make the autocorrelation function, it looks kind of like the point spread function in the microscope. So it's really just showing us that what we're looking at is sub-resolution, and we don't have any dynamic information. But what Enrico realized was that if you take an image of something that's moving very fast, you actually get an autocorrelation function that is only showing correlation along the x-axis. So if you think of the laser beam scanning across here, it comes across a molecule and you get an intensity peak. And then it keeps scanning, comes back, moves down a line and comes back again. And when it gets back to that location, that molecule is not there anymore. And so the correlation function can pick this up from thousands and thousands of images. And we were able to actually quantify protein diffusion in the cytoplasm of the cell from just regular confocal images. And I don't think I have it here because these are older slides, but I have a movie of just GFP in solution, and it just looks like noise. You know, I say for those of us from the pre-digital age, it looks like your cable went out and you've got that staticky screen and you pull out these kind of functions. It's just tremendous. So this technique is quite broadly used now and implemented in a lot of the commercial software packages. So again, at this point, we were working on a very simple confocal. It was an Olympus FlowView 300. It was pretty manual. I didn't have to put the filter cubes in and out, which was kind of nice. But everything else was manual. The pinhole adjustment was with screwdrivers, and the excitation emission filter reels were on sliders. And we actually customized ours with a GFP cube to make it really sensitive. So rather than using the 2-color cube, we used a single cube. And you can see we had this high-end computer monitor on the system, and it was quite a workhorse. So towards the end of my postdoc, I was looking to move back to Canada, and this job came up at McGill to run a facility. And I kept getting in trouble in my postdoc lab because I was helping everybody with their projects, and I was letting people from other labs come in and use the microscope. And my boss was like, "What are you doing? You're supposed to be working on your project." So when this job came up, I thought, oh, wait a minute, maybe that would be good for me because I like helping other people with their projects. And it was in Canada. It wasn't quite close to my family, but closer than Virginia. And so I took the position as the imaging director, and at the time it was just called the imaging facility. And it was an interesting decision. I had a lot of people really tell me that I failed. Like, what are you doing? Why are you not pursuing a PI position? And I even had people tell me, you know, you can do it, you know, you could really do it. And I'm like, yes, maybe I could, but it's not what I want to do right now. So it was interesting to get the different feedback from different people. And I think from a pure academic standpoint, some people really had seen this as a failure. And at the time, there weren't that many facility directors either. It was a fairly rare kind of job. And then I was really reflecting to myself too, like what is my impact on science? If I impact 100 people coming to use my facility in a little bit of a way so that they get better imaging for their science, is that equal to me training 10 really high quality PhD students who go on to make their own labs? And in my opinion, you can't really compare them directly. They're both important. And the best advice I got was from one of the other PIs at University of Virginia, who said to me as I was leaving, I think if the facility hires somebody really good, it will be successful. And so that was kind of a validation from the same sort of community, but a different viewpoint. So at that time, we had confocal, wide field, spectral imaging, and FCS. I was going from a FlowView 300 and some basic wide field scopes to 4 Zeiss 510 metas, which was the state of the art at the time. I was a little disappointed they hired me after they bought the equipment because I don't think I would have bought 4 identical microscopes for the institution, but they were really the state of the art at the time. We also had some more basic confocals, which were the Pascal 5, but again, very much more advanced than the FlowView 300. Some wide field scopes, and then still my favorite scope, the Axioscope. This is a fully manual upright microscope, which we're still running on Windows XP, I think, and has color camera and a black and white camera with the same chip. So it's really good for teaching about bare mass and cameras. And one thing I did find right away was that the 510s were much less sensitive than the FlowView 300 because they were designed to do so many different things. There's many more optical elements. So even though it was had much more capabilities, the sensitivity was a bit lower. And I went from a lab that was kind of doing cutting edge live cell imaging to a lab that was, to a institution that was doing mostly fixed cell work and wanted pretty pictures. So I'd kind of gone back to where I was with my graduate work, trying to convince people to do things that were a bit more quantitative. So I've been in the facility now for 17 years. I currently have 3 full-time staff, Bill, Joel, and Barbara. And I have 2 staff that are currently on leave. So Nellie will be coming back in February. We have 250 users. We have our little motto from planning to publishing because we really try to convince people to come before they start their experiments. 120 labs from 4 faculties. We have a few companies using the center. We do focus on live cell imaging, so we really try to encourage people to do that, and training, education, courses, and workshops. And I won't go through our whole list, but we have an array of different types of microscopes, and we're also managing microscopes for the Department of Biology and the Goodman Cancer Institute. And I just wanted to point out that our Zeiss Axioscope is still in operation. So that's the only microscope from when I started that. So that microscope's probably about 20 years old because it was there before I arrived. So for training and education, we offer a lot, probably similar to a lot of the facilities people here are working in, with consultations and training. We do have a step-by-step program that we developed during the pandemic where we actually walk through PowerPoint slide by PowerPoint slide exactly how to use the microscope. And we've really found that helps with training and consistency so that all the staff are giving the exact same training. And we also set it up so they can do the whole training remotely. So during the pandemic, when it was difficult to even have 2 people in the room, we could run through the whole training remotely. Even if you went in the office and the person was at the scope and you were down the hall. We do Illuminate seminars. We're just planning for our September one, where it'll be in person again, and we'll have a little wine and cheese reception to network with our users. We've set up a national Amiro instance in the cloud that we're now just trying to get set up a workflow so that we can get other researchers to put their data there. And the Montreal Light Microscopy course we've been running since 2010, but we haven't run one since the pandemic. So we'll be running that again in March. So I was thinking to myself, it'll be right around the same time of year. It'll be just towards the end of winter in Montreal, but the average daily temperature is probably about -10 Celsius. So I don't know if anybody from here would want to come, but we have nice, well-heated buildings. So and tunnels underground downtown for shopping. So, and then just recently we've come up with a comprehensive course on research data management, which is available on the Global Biomaging website. So at this time, I had been in the facility for about 5 years, and the grant was running out, and nobody except me seemed to be worried about where my salary was going to come from. And so I pushed I had some advocates. So for any of you who are early career, if you can really try to find a couple of advocates in your institution who will really support you and fight for you. I had 2 people like that this time, and you know, I explained the situation to them, and I told them I'm looking around for other work because I don't know what's going to happen to the facility, and that they were able to go to bat for me with the university, and I was given a assistant professor position. So it was a bit rare because I had already been there for 5 years. I was about I guess 13 years from my graduation from my PhD, and just starting my own lab. And the nice thing about that is the salary in Canada, the PIs are paid by the university. So my salary was secured by the department. And then I was able to bring in students and supervise students and also write my own grants. So it really brought me a lot of autonomy, but I still kept everything really aligned with the facility. So my research, I have listed some things here. Continuing the work on adhesion and cell migration, microscopy standards, and so on. And but it all really overlapped with the facility. So I kind of have these 2 hats now, but everything kind of goes back and forth between the two areas. So I'm just going to give you a snapshot of a couple of projects in the lab, and then wrap up with how I got involved with the networks. So when I got to McGill, as I was mentioning, most people were doing fixed cell, just looking for images. So I was really excited once I got my own funding just to get back into the correlation microscopy. So this is a project by 2 master's students who worked with me, Colton Boudreau and Abira Raja. And we went back to looking at paxillin and the different adhesions across the cell. So there's these small dynamic adhesions at the front, nascent adhesions, focal complexes, and more larger stable focal adhesions. And we wanted to understand the differences in these different populations. So paxillin is what's called an adapter protein in this layer of the adhesion near the membrane. And the dynamics are quite rapid. So we applied now image correlation microscopy, but in time. So we did these experiments on a turf microscope, and we collect images very quickly so that we can actually do an FCS-like experiment, but at every pixel in the image. So we basically look through time at each position in the image. And using software that was developed in Enrico Berton's lab, we were able to calculate the binding kinetics at each pixel. And we did a little bit of averaging. The data is quite noisy. So we basically went with short, intermediate, and long binding times. And you can see in the protruding edge here, the dynamics are a little bit faster. In retractive edges, they're a little bit slower. And the thing that we were most excited about was when you zoomed into areas where the cell was protruding, it seemed that the more dynamic part of the adhesion was always near the membrane, and the less dynamic was further back. And this fits well if you look at the actin cytoskeleton there, that this area of the adhesion is much more stable than this area out here that might be building if the cell is protruding out this direction. And the interesting thing was that there was a reversal of this when the cell was retracting. So the cell's retracting at this part, and you can see that the higher dynamics are on the inside. And you can imagine if the cell's retracting that the adhesion's rebuilding on the opposite end. So we went back and then applied image cross-correlation microscopy to the same system, now looking at paxillin and PAC, which is a focal adhesion, or sorry, a kinase that phosphorylates paxillin and activates it. And you can see from these images, it's kind of reminiscent of the integrin, that it's not very clear what's going on if it's really localized. You can kind of see it a little bit in some adhesions, but it's quite diffuse. So we did image cross-correlation now, looking where paxillin and PAC were binding together. And it really matched up with the fast dynamic part of the adhesion. So here you can see an area where the cell's protruding, and you see this really rapid dynamics along the edge near the adhesion, or sorry, near the membrane edge. And then when the cell's retracting, you see that same reversal. So this really showed us that that fast binding was related to the paxillin being activated, and that it was in these subdomains within the adhesion. And we were able to show that it was dependent on the kinase activity. Whoops, sorry. It was dependent on the kinase activity of PAC. So we used a drug inhibitor, and we saw these red areas, are showing where they're co-binding as a complex. And you can see when we treat with this drug for 60 minutes and deactivate the kinase, they're not binding together anymore. But they're both still in the adhesion. So you can still measure the dynamics of each, but they're not binding and unbinding as a complex. And then, as I always teach in all of my courses, and I'm sure Lionel does too, controls, controls, controls. So with cross-correlation, I'm always paranoid that we're just picking up an artifact of crosstalk between the channels. So we made a paxillin with the two fluorophores on it on either end and see correlation everywhere. And then we found a protein tensin, which is localized to adhesions but doesn't bind paxillin, and we were able to show no co-binding there. So we were pretty confident our results were real. So I have another project on LPP, a breast cancer cell migration project. And I had graduate student Alex Kipas and then another graduate student, Alex Nowakowski. And they were both just superb students. So I kept joking that I have to find Alex's of Polish descent for my lab because they seem to do a good job. And I'll just give you a brief overview here. I don't want to go over on time, but LPP is a protein that my collaborator has discovered is important for breast cancer metastasis. And when they knock it down, the cancer doesn't metastasize. So we're really interested in how it's working. And it's found in this actin regulatory domain. It's in the Zixin family. So it seems to be linking the adhesion to the actin cytoskeleton. And when we knock down the protein, we see that the cells move much more slowly. So it's a little bit, again, it's hard to tell from the movies, but when we do quantification, we treat the cells with a growth factor, which is known to play a role in breast cancer. And in the wild type cells, they speed up, but when the LPP protein is knocked down, they don't respond to the growth factor at all. So we wanted to dig a bit deeper into the quantitative aspects of this. So we wanted to look at the adhesion dynamics. So we developed a workflow where we use Amaris to isolate the adhesions and track them and measure their intensities over time. And we specifically looked at protrusive areas of the cell. And then we can quantify the assembly and disassembly rates at those locations. And so Alex Kipas developed a developed a MATLAB script, which would basically detect when it's disassembling, when it's stable, and when it's assembling. And we were able to do that across the adhesions. And what we found again is that when we have the endogenous protein without any mutations, that the adhesions are more dynamic following growth factor treatment, but when we knock down the protein, they're not. And the last one I just wanted to show, I just love these experiments because they're super hard, but the results are so beautiful. So this is a tension sensor for, in this case, vinculin, and it's based on a little piece of spider silk here that is an elastic molecule. It's a spring, I guess. And when the molecule is relaxed, the two molecules fret, so you get transfer of energy from the teal to the venous. But when the molecule's under tension, that spring separates, and you don't have that transfer anymore. And so we were able to apply that in cells with the LPP and the knockdown. And here we're showing that when you treat with growth factor, there's more tension across the adhesions. So somehow that the LPP is regulating the tension across the adhesions. And we did similar experiments with a tail-in version of this, which is GFP and tag RFP. And we did the whole gamut of experiments where we see when we have the LPP knockdown, we don't see this decrease in fret or increase in tension anymore. So I had a couple of other, so for microscopy standards and quality control, this is something I've been interested in from the time I started in the facility. And I was going to make a plug for two things. We just published a preprint of a 3D tissue standard for looking at microscopy quality control. And then just in August, I did a seminar with Lionel for the Latin American community, and that seminar is available on YouTube. So if you're interested in hearing more about the standards and quality control work we've been doing and others have been doing, you can see that. And we also, I've always been really interested in live cell imaging from my time in Virginia, of really understanding how the light can damage the cells. So this is a nice project also by Alex Keypass from my lab, where we looked at illumination overhead, which is basically extra light getting on the sample due to how the microscope is set up. And we were able to publish some phototoxicity assays and also a nice workflow of how to determine the best sampling time for live samples. So this is just the last example. It's a quick one, and we've just been getting interested in applying AI machine learning algorithms. So we're not interested in developing new algorithms, but we want to bring those tools to the biologists. So we want to have workflows to test them out and see which ones are ideal for different applications. And in this case, we were using what's called an adversarial network, or a GAN network. And we were generating synthetic images of actin from adhesions. So this is the real image of Paxillin adhesions. This is the real image of actin. And then we're able to train the AI algorithm to generate these synthetic images of actin. And the idea here is for live imaging, you'd be able to predict what the actin would look like without having to put in that second probe and have that second imaging workflow. So I'll wrap up now just by talking about the national international networks, which I think ties in really nice with the meeting. And there was interest in Canada of forming a national network of core facilities so that we would have a bigger voice to the stakeholders and the funders to have something similar to actually what we see in Argentina this morning, which is really exciting. And I had heard a talk about Eurobioimaging at the Association of Biomolecular Resource Facilities meeting in the state. And I reached out to Antje Kepler, who you just heard from, and Federica Pania. And this was the image on their website for Eurobioimaging. And I saw this big arrows going across the ocean to the United States. And I thought, oh no, I have to get Canada in on this. So the minute I started talking with Antje and Federica, I knew that I wanted to work with them. They seemed so wonderful and really wanting to help. And so I got involved with global bioimaging around the time they were getting started, around 2015. And from that, we formed the Canadian Network of Scientific Platforms, which was more like the example from Argentina this morning, where we are crossing all technology areas. So it's not just bioimaging, it's anybody running an infrastructure platform in a university. And we really see that group as sort of the hub in between scientific infrastructure researchers and trainees. So, we're training hundreds and hundreds of the next generation of scientists in all these technology areas. And we're currently working with the helium, a group in Canada and NMR on the helium crisis to try to write a position paper for the government to make some changes there to deal with the helium shortage. And as part of that, we created, so this is just showing the breakdown of the organization, 45 institutions, 195 or 94 platforms and so on. And we created Canada Bioimaging as a technology node underneath that umbrella structure in order to link with global bioimaging. And we're doing some of the very similar things that Ansha talked about, trying to exchange experience and build the community. So I apologize, I'm going a little bit faster, but I want to leave a few minutes for questions. So when Biomaging North America was forming, the group that was looking into it in the US approached me as the Canadian representative for the group. And I realized that, the work I was doing in Canada and the work I was helping global bioimaging with was easy to translate to North America. And so I agreed to be part of that original leadership team. And I've learned so much from them. And I hope I'm bringing, our experience from Canada and my experience with global bioimaging as well. And there will be a detailed BINA talk and video available at the GBI meeting if you want to hear more about them. It'll be available on the YouTube site after the conference. So Biomaging North America is broken down into working groups that focus on different areas that the community is really interested in: communications, training and education, diversity and inclusion. I'm involved with the quality control and data management group. There's a corporate partners group and imaging informatics. And so it's really built up by the members running these different work groups. So Canada Bioimaging is basically part of a network of networks where it's under the umbrella of the CNSP in terms of sort of the more overarching advocacy, professional development, sustainability aspects, and then part of Bioimaging North America with more of a technology microscopy community building. And now the global bioimaging map nicely has Canada up here and the Bioimaging North America, and of course Latin America now as well. So I think I acknowledged most of the students in my lab as I was going along. I apologize that this is my sort of research acknowledgement slides. I don't acknowledge all the people from the networks who've been really involved with that. But a lot of them are here or listening. And I'll thank you and take any questions.
Well, thank you very much, Claire. Very motivating talk. As far as I know more about you, I can believe how much coincidence we have, because I also begin doing electrochemistry when I did my undergraduate. It's unbelievable. Anyway, so there is a lot of coincidence. And also, I wanted to make a point about what you say of how difficulties in our career always bring some other opportunities. And I have a very short career, but when you have difficulties and you have always positive thinking, you end up having or being in a better position where you are. So I'm really thankful for the talk. So there is time for questions.
Thank you very much for your talk. It was very inspiring. Actually, I was wondering if you can give us your thoughts regarding how scientific institutions, particularly nationwide, should evaluate the positions of directing this type of facilities. It seems to me that at least in my country, in Argentina, there's a kind of established way to evaluate the career of researchers, the careers of also of technicians, but somehow recruiting and allowing people to grow as managing facilities still not established. And this is simple but important reason it's stopping us to grow into these very important positions to be filled with scientists. Scientists, seems to me that they are currently being a managing director on top of their day job as a scientist.
Yeah, so I completely agree with your assessment. And I think the main challenge is it's a change of culture. So people are used to measuring numbers of papers, number of students graduated, number of grants, and they don't know how to put a number on what we do. And so we're trying to develop some tools in order to measure, start to keep track more of like how many trainings has somebody done on their microscope and how many students have they trained. So my staff train about 200 people one-on-one per year. You know, what's the value of that to the institution? And I don't think it's really understood by the upper management. So I think my first business when I go back is going to be to take them for a tour, as we were recommended, and talk to them about how many students are impacting. So I think we need to come up with new metrics and indicators of the impact. And I think that's going to be a big part of the focus of the GBI meeting coming up on Wednesday. Just finding new ways to transmit that. And I think a lot of the same issues you see with open science, right? People don't understand what's the value of doing that. And if you put a code up there and 1,000 labs use your code, what have you given to science? How do we measure that? So I think it's going to take time because it's changing the way people think. But I think it's something that these national and international networks can really help to tackle together as a community.
Thank you very much.
Any other question?
Hi, very inspiring talk. Thank you very much. Thank you. I have the impression that I'm very, very interested about the key concepts that one should take into account for data management. I have the impression that the standards that are normally set by the grant agencies individually on every investigator. So what do you think are the actions that we need to take in order to set the standards from the imaging facilities?
Mm-hmm. So Global Bioimaging published a recommendation about international recommendation for research data management. So I would recommend taking a look at that. But I think this falls again at these communities. If we can really come together and come up with recommendations and workflows and present them to the researchers and also to the funders. So Canada's in the same situation where they've just come up with this policy on data management, but there's no funding. So they're telling everybody what we need to do, but they're not actually providing any resources for the hardware or the human resources. So again, I think people think I'm going to put my data on a hard drive and I'm going to put it on, in a file cabinet. They don't think of how much work it takes to really manage the data, to share the data, to annotate it and curate it. And so I think, again, it's educating the people who are in charge, of the funding, to let them know how much work this is, but also how much value if we really do it properly. And it's not just putting it on hard drives and handing it to your collaborator, So I think it's a huge challenge we need to face. And we just got a lattice light sheet. So maybe I'll have more information for you on what we've come up with, but it's definitely an ongoing challenge. Yeah.
So you mentioned in your talk about this, this struggle with sort of roles and a sort of self-identification. So this when you said I want to go manage a facility, and there was this attitude of why, this is like you're turning your back on academic structures. That's still a very prevalent problem in Latin America. Well, I'm extending from Mexico, but I mean, there's still those sort of attitudes which I guess you're facing maybe sort of 15, 20 years ago are still very, very current here. We're not so far down that line. That decision you made to then restart your research program, did you meet resistance? Was that just a personal decision you took and then pushed for, or did someone invite you to that? Or because we still put things in boxes, we still say, well, that's a core facility, therefore they do service, and if you want to do research, that's what PIs do. And I don't see why, I think any core facility that is being blocked from doing sort of research or is, and it happens, is such a waste. I mean, who benefits? It just, it requires a change of mentality, it requires a change of, a sort of more flexible mindset, but there is resistance to that. So I was just wondering that process for you to restart your research program, which is wonderful because the research you were doing was really, really high end and fantastic, and it's contributing really well. Yeah. Did you have resistance there? Was there voices against that?
Yeah. So for me, it was completely my own initiative for my own sustainability, right? Like the facility was going to end because there was clear that the fees were not going to cover all of our expenses. So that was the main goal. But the second goal was that I was finding the research projects in the facility weren't really taking advantage of quantitative imaging and the advanced tools and, you know, things like these FRET sensors. And I didn't have time to develop them as the facility director. But if I had my lab and could get grants and have students, then I could get back to developing those things. So those were kind of the two forces that were driving me. The only reason it was successful is because I had those two advocates, and they went and advocated for me, and they were well-established professors and chairs of departments. And I don't think it would have happened without them. I wouldn't say I had resistance from others because they were able to be creative. I didn't take a seat, like I didn't take a faculty position from a department. They found a creative way to find a position for me. But on the other side, people treated me differently in a positive way, which really bothers me. Like the same people I was working with the week before now treated me like a professor instead of a facility director. And, you know, they would say hi in the hall, whereas the week before they didn't. And I was going to the same office, the same job. So I agree with you. I think we still have a lot of work to do. And we need to show the value of these positions in order for people to really appreciate the impact we're having on science. And it's not trivial, but I think it is changing though. And I think especially the younger researchers, they come in, they're like, where's the facility? Who do I need to talk to? And they really, really appreciate the help they get when they're starting up their labs. So I am hopeful that it's shifting, but again, I think it'll take time.
Yeah, I mean, institutions resist change. I mean, that's what they do. And so,
yeah,
and people can start to feel uncomfortable, I find. And so you're not in the normal the categories that they've been used to for 20, 30 years. And it can be, it can create resistance.
I had colleagues say recently, it's like steering a really big ship and trying to make a quick turn, you know, they've just been going that direction for so long that it's going to take a lot of energy to put it on a different course. Yeah.
Thank you, Chris. Well, thank you very much, Claire, for a wonderful talk.